By the 1960s, a critical gap had emerged. The global population was growing exponentially, but the amount of land available for farming remained fixed. Closing that gap required agriculture to evolve into three distinct strategies: plant breeding, disease and pest resistance, and biofortification. Scientists like Dr. Norman Borlaug and Dr. M.S. Swaminathan addressed this by manipulating the genetic makeup of crops to create high-yielding varieties. This shift initiated the Green Revolution, pushing wheat and rice production to levels that met the demands of a surging population. At its core, plant breeding is the purposeful manipulation of plant species to create genotypes and phenotypes that are better suited for cultivation and human benefit. Modern agriculture functions as a system of high-precision genetic management, designed specifically to secure human survival. Conventional plant breeding follows a standard 5-step flowchart. Step 1 is the collection of variability. This involves gathering a crop's germplasm, the entire collection of diverse alleles from all wild varieties and relatives. Step 2 is the evaluation and selection of parents. We screen the germplasm to identify specific plants that possess the desired traits, like high protein content or superior yield. Step 3 is cross-hybridization. To combine traits from two selected parents, we must control the pollination process. This requires emasculation, the manual removal of anthers from the female parent, followed by bagging to prevent the stigma from being contaminated by unwanted pollen. Step 4 is the selection and testing of superior recombinants, where hybrids are self-pollinated for generations to reach homozygosity. Step 5 is commercialization, involving multi-location field trial that naturally exist in sexually compatible parents. To bypass limits, we use mutation breeding, altering DNA with gamma rays, and somoclonal variation via tissue culture. For instance, mung beans gained yellow mosaic virus resistance through mutation breeding. However, the okra variety Parbani kranti gained this resistance through conventional hybridization with a wild species, not mutation. Each strategy, conventional, mutation, or tissue culture, is selected based on where the specific survival gene is found. Pathogens like fungi and bacteria destroy 20 to 30% of crops. This matrix lists the specific resistant varieties you need to master. Himgiri is a wheat variety bred for leaf and stripe rust resistance, while Pusa swornum is a brassica resistant to white rust. Plants also require protection from insect pests. They utilize morphological defenses, like hairy leaves and cotton to repel jacids, and biochemical defenses, like solid stems in wheat to resist the stem sawfly. Our breeding programs replicate these defenses. For example, the brassica variety Pusa Gaurav was specifically developed to provide resistance against aphids. Let's test your recall. Look at Pusa Komal, Pusa Shubra, and Pusa Sawani. Match these varieties to their target crops and threats. Building these defenses ensures the crop reaches maturity, but the final step is ensuring that the harvest provides actual nutrition. Over 3 billion people suffer from hidden hunger, a chronic deficiency in essential micronutrients like iron, zinc, and vitamin A. We address hidden hunger through biofortification. Atlas 66 is a wheat variety bred for high protein. We've also developed iron-fortified rice and maize hybrids with doubled essential amino acids. Furthermore, AARI released biofortified vegetables, including vitamin A-enriched pumpkins and vitamin C-enriched bitter gourd. Biofortification shifts the focus of agriculture from delivering simple calories to providing essential public health nutrients. Here is a final exam scenario. If an okra crop is devastated by yellow mosaic virus and no cultivated varieties possess resistance, what breeding strategy is required? Since the trait is missing from cultivated varieties, the resistance gene must be transferred from a wild species through conventional hybridization, creating a cultivar like Parbani cranti. Screenshot this summary. It combines the 5-step process, the resistance matrices, and the core biofortified crops into one revision sheet. Understanding these strategies is the key to mastering the science that maintains the global food supply.